Welcome to the cell division series. This video focuses on chromosome structure. At the end of this video, students should be able to understand how chromosomes become compacted into the nucleus of a cell. Chromosomes are tightly coiled and compacted units of heredity that hold our DNA, or blueprint, for the organismal development. Humans have 46 total chromosomes, or two sets of 23 pairs. One set from mom and one set from dad. This karyotype shows that there are two copies of each chromosome. So chromosome number 1 shows one copy from mom and one copy from dad. Chromosome number 13 shows one copy from mom and one copy from dad. This continues for each of the 22 somatic or body chromosomes. And a copy of chromosome from mom and copy of chromosome from dad for the 23rd pair, the sex chromosomes. Each eukaryotic human chromosome can be hundreds of millions of base pairs in length, all needing to be compacted into the tiny nucleus of every cell inside of our bodies. In its relaxed state, DNA and its associated histone proteins are called chromatin. Our DNA wraps around these proteins to begin to condense in order to fit into the nucleus. There are 8 histone proteins that our DNA wraps tightly around, twice, making up a nucleosome. One additional histone links the neighboring nucleosomes together, making a chromatosome. The nucleosomes shorten the DNA 7 times more than its original state. As the nucleosomes condense on one another and coil into a solenoid, further condensing and zigzagging onto itself, it forms the 30 nanometer fiber. The fiber undergoes radial looping, held together by SMC proteins, to continue compaction. These compacted forms of DNA are still loose compared to the chromosomes that are ready to undergo cell division. This relaxed, loose form of chromatin is called euchromatin. Remember, when you are relaxed, you chillin', euchromatin. This is the form that undergoes transcription for protein synthesis. The heterochromatin is more tightly compact. Typically, the euchromatin has the genes, which makes sense since the transcription can take place there. Higher compaction would happen in non-coding regions like the telomeres, or the ends of the chromosomes, and in the centromeres, which are used during cell division. During interphase, the cell is just making the proteins it needs to survive and get ready for division. So under the microscope, it looks like a dark blob with tiny dots inside. The blob is euchromatin. The dots are the heterochromatin. During division, the chromosomes condense even further by supercoiling into highly condensed versions. We can see stained chromosomes under a microscope. These supercoils are what we recognize as the X-shaped structures of the chromosome in prophase of cell division. Students should be able to contrast euchromatin and heterochromatin. Students should be able to identify stages of chromosome condensation. Students should be able to explain the difference between chromosomes and chromatin.